Hello there. Once again, we want to look at another requirement of life, which is digestive system. Earlier, we have mentioned that body systems are important for life because they supply materials to cells, tissues, and organs, and at the same time, they remove other materials like waste from cells, organs, and tissues. Another important system that multicellular organisms require is digestive system. So what is a digestive system? Digestive system is a long tubular structure running from the mouth to the anus. I have a diagram here to illustrate how a digestive system will look like. So this part is called mouth, and then it runs, it's a tubular structure running down as it can be seen up to the terminal part, the anus. Digestive system is made up of organs. From the diagram, we can identify some of the organs. I've already identified one part there, the mouth. So one part of digestive system is the mouth. Down the mouth, we have another part called esophagus. Esophagus. Another common name is called gullet. And then down it will run into another structure. We have the sphincter muscle. Sphincter muscle. Then the stomach. And then another sphincter muscle, we shall give them exact name. We have the liver, close to the liver we have gallbladder, and then we have the pancreas. And then from the stomach, it runs into another structure, small intestines. Which connects to large intestines. And then the lower part of large intestines, rectum, and the very last bit, the anus. But we have a vestigial structure there called the appendix. So those are the organs. But it also, we can identify some accessory glands. Accessory glands, although they cannot be seen from the diagram, but it also has accessory glands, including in the mouth, we have glands called salivary glands. In the stomach walls, on the stomach walls, we have glands called gastric glands. Gastric glands. So we are going to look at each of those organs and find out their role in digestion. First part is the mouth. So in the mouth, if you look at mouth of any human being, we have structures like teeth or organs. The role of teeth is for chewing, simple chewing, or biological term mastication. So what is this chewing or mastication? Simply breaking down of large complex food materials into small soluble forms that can easily be absorbed. That's why you chew. You chew first of all to increase surface area for digestion, and then you chew also to break them or to break food particles into small pieces so that you can easily swallow. And then we have tongue. Without the tongue in the mouth, it will be very difficult for swallowing. So tongue has two roles. One of them is turning food during chewing, and then also helps in swallowing. For you to swallow food, the tongue will push the food backwards. In the mouth also, we have mentioned a very important gland, salivary gland. There are several. We have salivary glands. One of the glands is called the parotid gland. Parotid gland. Parotid gland is located in the upper part of cheeks. We have sublingual gland. Sublingual gland is located below the tongue. And then we have submaxillary gland. Submaxillary gland is just located in the lower part, the lower jaws, at the back of the lower jaws. So the role of salivary gland is simply to secrete saliva. Secretes saliva. The role of saliva include 1. Softening of food. Softening of food is important because you cannot swallow dry food. It's going to choke you. So it has to be softened by use of saliva. And then another role, it dissolves food. It dissolves food. When you put crystal of sugar into your mouth, they are not going to remain in solid form, but they'll change from solid to dissolved form because saliva contains water. And then again, the role of saliva is to lubricate food. Lubricates food. Lubricates food. Why should food be lubricated? Food is supposed to be lubricated so as it can easily be swallowed to reduce friction between the food boluses and the lining of the alimentary canal. And then the very important role of saliva is that it contains enzymes. Contains enzyme. The major enzyme found in the saliva is called amylase. Amylase enzyme. So what is the role of amylase enzyme? Amylase enzyme usually acts on starch and converts it into maltose. Converts it into maltose, which will be further digested in another part of the digestive system. So after the food has been chewed in the mouth using the teeth, using the tongue, it will be rolled into small balls called boluses. So boluses will move down the alimentary canal or digestive system by a process called peristalsis. So where does peristalsis take place to allow swallowing? It takes place in our esophagus. It takes place in the esophagus. So the role of our esophagus is to allow movement of food from the mouth to the stomach. How does food move? We have mentioned that it moves by a process called peristalsis. 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 So what is this peristalsis? Peristalsis is a series of rhythmic contraction. Contraction of muscles found in the esophagus. So by peristalsis, food is able to move down the esophagus. You can be able to feel peristalsis yourself even if you swallow any kind of particle. Let it, even if it is saliva, it will go down. You'll be able to feel how it is moving. So peristalsis, apart from just the contraction, also the food will be able to move down the digestive system by aid of gravity. Although sometimes even at if, for example, you turn your body upside down, still you are able to swallow because of peristalsis. We have another very important organ, the stomach. But before food reaches the stomach, it will go down the esophagus, as we have said, by peristalsis, and it will encounter a valve-like muscle before it reaches the stomach. That valve-like is called the cardiac sphincter muscle. So for food to enter into stomach, this cardiac sphincter muscle is going to relax. When it relaxes, it allows food to get into the stomach. In the stomach, presence of food will stimulate some reaction to take place. Presence of food stimulates production, stimulates stomach walls to produce gastric juice. Gastric juice. This gastric juice has several contents. Some of the content in gastric juice includes enzymes. We are going to find out the enzyme. Include mucus. Include hydrochloric acid. So it will have stimulated gastric glands to secrete gastric juice, which contains enzymes, mucus, and hydrochloric acid. But we have specific parts of gastric glands that secretes each of those composition. For instance, we have cells in the glands called chief cells. Chief cells. Another name for chief cells is peptic cells. Peptic cells in the gastric glands are the one that secretes an enzyme. That enzyme is in its inactive form called pepsinogen. Pepsinogen. This is an inactive form. The role of pepsinogen is to break down proteins into peptides. But before it acts on proteins, it will have to be converted into active form. The active form of pepsinogen is called pepsin. So how is pepsinogen converted into active form pepsin? Hydrochloric acid comes into place to convert pepsinogen into pepsin. So we have cells in gastric glands called parietal cells that secretes hydrochloric acid. Secretes hydrochloric acid. So we have said one role of hydrochloric acid is converting pepsinogen into pepsin. That's the major role of hydrochloric acid. But apart from that, apart from converting hydrochloric acid into pepsin, another role of hydrochloric acid is providing correct pH for working of pepsinogen. Because pepsinogen, or pepsin being an enzyme, it requires an optimum pH. That pH is provided by hydrochloric acid. Another function of hydrochloric acid, sometimes you realize that you have taken food, you have ingested food, but maybe it was not to correct hygienic standards. So because of that, it could be contaminated with microorganisms. So it will be swallowed into the stomach, but most of the time you will not get sick. Why? Because hydrochloric acid, being of extreme pH, will kill the pathogens, and in the process prevent you from getting infected. So it kills pathogens. Swallowed together. Pathogens swallowed together with food. And then on the stomach walls, we also have cells called goblet cells. The role of goblet cells is to secrete mucus. Mucus is important in digestion. As we have mentioned, in the mouth, it lubricates food. But in the stomach, the major role of mucus is to prevent hydrochloric acid from destroying the stomach walls. From destroying stomach lining. Sometimes it has happened that there's not enough mucus, or there's excess hydrochloric acid being produced by the gastric glands, especially if somebody is stressed, or if somebody goes without food for a very long time. You'll realize that HCl will start to eat or to attack the stomach lining, and in the process one can develop ulcers. But most of the time it does not happen like that, because we have a protective lining in form of mucus. But now, what is the role of stomach? From what we have just mentioned, we can underline two roles. The major role of stomach is digestion. But of what? Digestion of proteins. We have already looked at it. The enzyme involved is pepsin, which is secreted in form of Trypsinogen. But apart from that, for storage of food, that's why when you eat, you realize that you will feel satisfied for some time. It stores food between 2 to 4 hours in preparation for digestion, for further digestion if they are not proteins. And also we have mentioned that it digests proteins. It forms the starch. That's where protein digestion starts. Then down the stomach, we have small intestines. Before food leaves stomach into the small intestines, there will be also mixing of food particles with the gastric juice. That process of mixing that occurs in the stomach is called churning. So churning produces food that will go into the small intestine in form of chyle. So what is this chyle? Chyle is that food that is leaving the stomach, getting into the small intestines, because now it is mixed with the gastric juice that contains the 3 components that we have already talked about. But for food to move down the stomach again, it is by peristalsis or peristaltic movement. In the small intestines, is divided into 2 major parts. We have a part called duodenum. Duodenum is about 0.5 meters long and forms the first part of the small intestines. The anterior part forms the anterior part of the small intestines. So what is important in duodenum? Duodenum, food is received from the stomach and gets into the duodenum. But duodenum usually receives some secretion from 2 places or 2 organs. One of them is pancreas. Pancreas secretes pancreatic juice. Pancreatic juice contains majorly enzymes. What are these enzymes? One of them is an enzyme called lipase, sometimes referred to as pancreatic lipase. Now remember, in your previous year you looked at naming of enzymes. Enzymes sometimes are named by the substrate that they break down. Therefore for lipase, it is coming from the word lipids. So from this, it can easily tell us that it is the one that digests lipids. So the role of lipase is digestion of lipids into fatty acids and glycerol. Digestion of fatty acids and glycerol. Another enzyme is pancreatic amylase. It is specified pancreatic amylase because in the mouth we are able to come across another amylase. We call that one salivary amylase. But they have the same function, only that their source of production is different. Amylase. Pancreatic amylase. Pancreatic amylase acts on starch. It breaks down starch to form maltose. Breaks starch to form maltose. Another enzyme is pancreatic trypsin. Trypsinogen. It's also secreted in its inactive form, trypsinogen, but it will be converted to active form, trypsin. It will be converted to active form, trypsin. So the question is, what converts this trypsinogen to trypsin? We have an enzyme that is secreted by the walls of duodenum called enterokinase. Enterokinase converts trypsinogen to trypsin so that it can now be active. So the question is, why is it that these enzymes, some of them are being secreted in inactive form? We have seen pepsinogen, trypsinogen. They're supposed to be secreted in inactive form so that they do not digest the glands and cells that secretes them. So what is the role of trypsinogen? Trypsinogen acts on proteins, again, to form peptides. So you have come across three enzymes in pancreatic juice: lipase, lipids to fatty acids; amylase, starch to maltose; trypsinogen, active form trypsin, proteins to peptides. They are being secreted by pancreas in form of pancreatic juice. Another important secretion is another important organ in relation to duodenum is gallbladder. Gallbladder secretes or releases bile. Bile has two roles. One of them, before lipids are digested, sometimes they are consumed in form of fats. But fats are usually large in size. Even if they are being chewed, still they coalesce together. Bile will break down fats into fat droplets, small pieces of fats. So that process of breaking down small pieces of large pieces of fats into small droplets, we call it emulsification. So if you have very large sizes of fats, they will be broken down into smaller ones, like this. So we call that process of breakdown of large fat droplets into smaller droplets as emulsification. Now, why is it important that it should be broken down into smaller ones? This one is to increase surface area. Increases surface area for digestion. So that now the enzyme lipase can be able to get into contact with at least nearly each of those particles. And another role of bile is to provide correct pH for working of pancreatic enzyme. Provides correct pH for working of pancreatic enzymes. We have mentioned pancreatic enzymes. Those enzymes secreted by pancreas in form of pancreatic juice. But now the role of gallbladder, it releases bile. The only role is to release, which means it is not the source of gallbladder is not the source of bile. Source of bile is majorly the liver. Liver releases bile or produces bile, and then gallbladder stores. This one produces bile, liver, and gallbladder stores. When the digestive system is stimulated, it is going to release now the bile, which will perform the two functions, emulsification and providing the correct pH for action of pancreatic enzymes. So before we go to another part, we can look briefly at liver, the role of liver in digestion. Liver is important because, as we have mentioned, it's the one that produces bile. But this bile will go somewhere else, the gallbladder, for storage. And then, apart from that, after digestion, food has been absorbed. For example, sugar. When you take a lot of starch, it will be converted into sugar. This sugar is not, sometimes if it is too much in the bloodstream, there will be a problem. So it will go back, it will go through the liver for regulation. Regulates sugar and other foods like amino acids. Before they join the rest of the circulatory system. So liver, production of bile, and then regulation of amount of foods in the blood like sugar and amino acids. Another part of small intestines is the ileum. Ileum forms the longest part of the small intestines, forming about 5.5 meters in length. And one characteristic of ileum is that it is highly folded. Why should it be highly folded? To increase surface area for digestion and absorption. Increases surface area for digestion and absorption. But in terms of digestion, we want to look at it, what makes it very important in digestion. So the walls of ileum also releases a juice. That juice is called succus entericus. Succus entericus is a juice, just like pancreatic juice, gastric juice, but it contains enzymes. It has enzymes. The role of enzymes is to complete the digestion. Remember, we had digestion in the mouth, digestion in the stomach, digestion in the duodenum. Now, the role of ileum here, it's acting as the site where completion of digestion takes place. But that one will require enzymes. Some of the enzymes that are found contained in succus entericus include enzyme sucrase. Sucrase, we have said that enzymes sometimes are named according to the substrate they break down. So from this word sucrase, we can guess the substrate. The substrate will be sucrose. So it breaks down sucrose into glucose, and fructose. Another enzyme is lactase. Again, we can guess, or we can be able to identify the substrate, the first part of this word, lactase, lactose. So it means it breaks down lactose into glucose and galactose. Lactose is found in milk. Another enzyme is maltase. Again, we can be able to get the substrate or the type of food that will be breaking down maltase from maltose. To take you back, remember in the mouth we had amylase. Amylase was breaking down or breaks down starch into maltose. But that's not the final or the simplest unit of starch. That is a disaccharide. That disaccharide will have to be broken further to form a monosaccharide. That is in the ileum. So maltase comes in handy to complete the digestion of starch. Starch was converted to maltose. Now maltose has to be converted to glucose. These are the simplest form of nearly all of them. Then we also have lipases. Lipases breaking down lipids into fatty acids and glycerol. We also have another enzyme called peptidase. To take you back again, we came across peptides. Remember, in the stomach, there was digestion of proteins into peptides. In the duodenum, there is trypsinogen, inactive form of an enzyme, trypsin, which converts proteins to peptides. For peptides to be, or for proteins to be completely digested, it takes place in the ileum, where we have peptidase. So from, again, this enzyme, it means we can be able to get the substrate. Our substrate here will be peptides. So the role of peptidase is to convert peptides into amino acids. Amino acids are the simplest form or the building blocks of proteins. So at that point, they can easily be absorbed. So starch has been processed or has been broken down. Proteins have been broken down. So ileum, apart from just digestion, we were able to mention two roles. Digestion, we have seen the enzymes doing that. Sucrase, lactase, maltase, lipases, peptidase. After they have been broken down into their simpler form, the way we are seeing them, amino acids, glucose, fatty acids, and glycerol, it has to be absorbed into the blood stream. They have to be absorbed. So as we sum up digestion in the small intestine, we can simply look at the roles. One, we have said digestion, completes digestion. It completes digestion. We have seen how that one has happened. We have seen peptides being converted to amino acids by peptidase. Maltose being converted to glucose by maltase. Then another role is absorption of digested food. Absorption of digested food. After the food has been converted from complex to simpler form that can be absorbed, absorption mainly takes place in the ileum. But how does this one happen, absorption in the ileum? Ileum has some structures called the villi. If you look at the walls, we have finger-like projections on the walls of ileum. Those are called the villi. There are very many. And because there are many, they provide a large surface area. They increase surface area for absorption. Increase surface area for absorption. Then again, that wall that separates food in the ileum, in the villi, and the bloodstream is very thin. Thin wall in the villi. So thin wall in the villi will reduce distance. Reduces distance for faster absorption. For faster absorption. And then again, very important structure of the ileum is that it is well supplied with blood capillaries. Blood capillaries. There are very many blood capillaries. Where we have many blood capillaries, it increases diffusion gradient. Diffusion gradient. In other words, food materials are going to be absorbed very fast because of many blood capillaries supplying blood and taking away the food materials very fast. So those are the major roles of the ileum. Digestion, complete digestion and absorption of digested food materials. But remember, not all food that is swallowed is going to be digested and be absorbed. Some of it will be undigested. Therefore, the food will move down into another organ called large intestines. Large intestines. In the large intestines, there is no digestion, but majorly we have absorption. Absorption of some few materials like water, mineral ions, and vitamins. Remember, we do not just have starch or carbohydrates being eaten. You eat starch, you eat proteins, you eat food rich in vitamins, mineral ions. Water is part of the daily requirements. They are not going to be digested, but they'll be absorbed directly in the large intestines. And then after that, again, we are going to have some materials being remaining. Remnants of materials include undigested food. Some materials will remain in the large intestines as undigested. You also have indigestible materials. Undigested means it could have been digested, but maybe it was not given enough time. Indigestible means, for example, seeds that have a very hard tester. So mainly large intestines is absorption. Then the last bit of digestive system is the rectum. In the rectum, there is storage of undigested and indigestible food materials. So in the process, these two shall form what you call feces. So in essence, in the rectum, there is formation of feces. There is formation of feces in the rectum. Then after these materials have stayed in the rectum for some time, and it reaches a point where it cannot be stored anymore, it will leave the digestive system through anus. So the role of ANA simply means lets out. Lets out feces. So those are the major parts of the digestive system. So as I finish, let us look at roles of digestive system as we complete. Roles of digestive system. We have mentioned two major roles throughout. One, digestion of food. We have seen digestion in the stomach, in the mouth. For example, starch to maltose. In the stomach, proteins to peptides. To duodenum, we had several materials being digested. In the ileum, we have completion of digestion. And then another one is absorption. Absorption of digested materials. So materials have, after being digested, they'll have to be absorbed into the blood stream. So this one summarizes everything: digestion and absorption of digested food materials. So as we complete, you can just look at this exercise. Complete the table. Below. We have substance. We have where produced. Where produced and role in digestion. So one of them is gastric juice, salivary amylase. We have bile and pancreatic juice. So this is what we have gone through, and we can easily be able to tell where they are produced and their role. Gastric juice. Gastric juice, you can remember in the stomach, we said gastric juice is produced by gastric glands. Gastric glands. The role of gastric juice, for you to be able to get the role of gastric juice, you have to mention, for example, pepsinogen. Role of pepsinogen. Pepsinogen. Role of pepsinogen is breakdown of proteins into peptides. HCl, hydrochloric acid. Role of hydrochloric acid, we have said, is the one that converts pepsinogen to pepsin. Pepsinogen to pepsin. We mentioned several. At your own time, you can complete. Then we have salivary amylase, where produced salivary glands. Salivary glands. The role of salivary amylase, breakdown of starch to maltose. We have bile, where released gallbladder. Gallbladder. Role of bile, emulsification. We can remember that term. Emulsification of fats. Breakdown of large fats into smaller fats. Pancreatic juice contains several enzymes and is produced in the pancreas. Released by the pancreas. Has several roles depending on the enzyme. So at your own time, look at the role of pancreatic lipase, and then role of pancreatic amylase, then role of pancreatic trypsin or trypsinogen. So I hope you have been able to enjoy the topic, and if you have any further question, we always provide our email. You can get in touch with us through the same email.